Okay. So this afternoon, we are going to look at the lymphatic system and I think it was infectious diseases. Let me see what the title was. Lymphatic, which includes infectious diseases. So that's what I've got on my deal. So this is a system that we really don't talk about in EMT school very much. We say lymphatic system, immunity, fighting diseases, and that's pretty much what we say. So, what are the functions of the lymphatic system? Well, the big one really is to return tissue fluid to the bloodstream. We'll look at this in a minute, but did you know that your capillaries are very leaky? The capillaries are leaky because that's the way that fluid and nutrients and waste products get back and forth between the cardiovascular system and just the interstitial space between the cells. So tissue fluid is lost fluid that if it doesn't get back into the bloodstream, your patient's going to have stuff like cankles. That's one of the big problems there. There's just tissue fluid outside of the bloodstream, outside of the cells that's causing the edema. The second function, you guys are already familiar with this. The lymphatic system pretty much protects the body against any kind of foreign invaders. Most of your lymphatic structures are located at key openings to the body and also potential entrances, such as the groin, the armpits, and the neck, where the trunk attaches to extremities in the head, basically. So, if you get an infection in your leg, for example, or you get a laceration or whatever, by the time the blood is returning to your heart, hopefully the lymphatic system will catch some of the bacteria or so forth in the groin area before it gets up into the trunk area. So that's why you see these, the location of the lymph nodes and lymph nodules. All right, so here's an overview. The lymph system here is in green. It does circulate. So it is a circulatory system like the cardiovascular system is, but without one thing, and that's a pump. So, whereas you have the heart pumping in the cardiovascular system, you don't have a pump here. So we'll have to talk about how does this stuff move around. So the components are going to be of the lymphatic system is lymph or lymph fluid, lymphatic vessels. They go from large to small, just like your blood vessels. Lymph nodes and nodules. The spleen and the thymus gland are the two other components. So if you look at this picture over on the left, you can see there's clusters of lymph nodes in the armpits, groin area, excuse me, in the neck. They kind of focus in those areas. All right, so we talked about the lost fluid. Because your capillaries are leaky, and in the picture here in red, you have an arteriole, which is a small artery. And then you have a capillary system here. And then to return the blood to the heart, you have a venule, which is a small vein. In this capillary system, now remember, capillaries are 1 cell thick. The wall is 1 cell thick. It's very thin. They're squamous, simple squamous cells. And yes, stuff can leak in and out between each cell. You have glucose leaking out. That's important. You have oxygen leaking out. That's important also. That's how these things get to the different cells. But you also do have a certain amount of fluid that's leaking out that glucose and oxygen and hormones and things are dissolved in. So we do have fluid, and it's out there in between the cells, and we've got to get it back into the bloodstream. Or like I said, you're going to have major edema. So lymph fluid is an interstitial, which means between the cells, fluid that's not returned to the capillaries, but we've got to get it back up to the heart, get it back in the bloodstream. It's similar to plasma. In fact, it came from the liquid part of plasma. There are some proteins in there and a lot of water for sure. So in short, I think this is a pretty good diagram because it shows you what happens to this fluid. So like I said, as the blood travels through the arteriole into the capillary system, the water leaks out here between the cells. All right. And then you see in green, these are your lymph vessels. These are dead end vessels. You see, they just start from nowhere and then they come together and create larger and larger vessels. Those are going to go back up to the heart. But these are just dead end vessels. And what happens is the fluid moves into these, that lost fluid moves into these and then starts traveling back up to the heart to get in back into the bloodstream. So that's what the very small lymph vessels do. So, like I said, how does it move? There's no pump to pump it around. So the movement of lymph fluid back to the heart is going to rely primarily on muscle contraction and body movement. I'll show you what I mean here in just a second. So it makes sense if you're more active, you will remove more of that lost fluid. I notice even with myself, if I start having some swelling in my feet, for example, because I sit at at a desk teaching for 8 straight hours with my feet hanging down, if I will go to the gym or if I will go for a walk, that fluid totally goes away and pretty quickly. So your patients who are sitting around all day, especially if their feet are not elevated, if their feet are just hanging down from their recliner or whatever, you can expect to see a lot of fluid there. And so that's why they need to elevate their feet or maybe get out of their recliner and go do something. That is not always possible with our chronically ill folks. So that's the unfortunate part. All right, we've already talked about the dead end lymph capillaries. They're in green in this picture right here. They uptake lost water and also some proteins. You actually have lymph capillaries internally in your intestine. And they're called lacteals. They uptake fatty acids and vitamin A, and that they're going to take them back to the bloodstream. So, you know, that's another good thing for our body. The lymph capillaries are similar to veins in that they have one-way valves. So as the lymph travels back to the heart, the valve behind it closes so it can't flow backwards down into our patient's feet or wrist or lower back or whatever. Now, as the lymphatic vessels get larger and start combining together, you will see the larger vessels do have smooth muscle walls. And as we know, smooth muscle can contract and push that lymph fluid back up to the heart. So those are the ways that it moves, since we don't have a pump. So pressure differentials are what is going to move the lymph fluid back into the lymph vessels. You have higher pressure here in the interstitial space because all that fluid has leaked out, and you have lower pressure in the lymph vessels. So that pushes by filtration, that pushes the fluid back into the lymph vessels. And over on the right, you can see a very close-up picture of the dead end capillary and how the water is getting back in there. Like I said, we're very leaky, and between those cells is where you have the leakiness. This is just another picture of how it works, the direction of flow, and so forth. And this is just another picture. This is a cross section. At the top, you see your arterial and your venule, and your capillaries are in pink. And then as the leaky stuff flows down here in green, I don't know why they always make it green. Here's your capillary, your lymphatic capillary, and the water going back in there. Okay. So your small vessels combine to make larger vessels, and eventually this lymph fluid is going to get dumped into your subclavian vein up there underneath your clavicle. That's what subclavian means, under the clavicle. And then it will go to the heart, be reabsorbed as plasma, pumped around, cleaned out, and so forth. So here's the funny part. And I tell you what, I looked everywhere last night to find out why, and I could not find a single reason. So if you look at the picture on the bottom, you'll see that lymph fluid coming from the right side of the head, the right shoulder, or the right chest quadrant, I guess, and the right arm. Those all drain into the lymphatic duct, okay, into the right subclavian vein. But then look in the light green. Everything in the upper left body and the lower extremities drains into the thoracic duct, duct, into the left subclavian vein. Why is it uneven? I couldn't find the answer for you anywhere. I've always wondered that, and I said, you know what, let me just look this up. One person mentioned, well, it might have something to do with the heart. The heart doesn't pump evenly, if you think about it, coming out of the right and left ventricle. So maybe this has something to do with the heart. I just found that interesting. So all of this lymph fluid ends up back in the veins, the subclavian veins, to the vena cava, back into the heart, and it kind of starts all over again. Here you can see, and again, they're green, the right lymphatic duct and the thoracic, where that lymph fluid comes back into the main circulatory system. In this picture, we see some lymphatic vessels in the arm and the breast, and you see a huge amount of lymph nodes here, because remember, it's at the join of an extremity with the torso, so that, again, if something enters the body, hopefully it can be gotten rid of before it gets into the main circulation. Now, a lot of times in breast cancer, the cancer cells leave the area and they get into the lymph nodes. The lymph nodes have white blood cells there to fight off and kill invaders, but sometimes they get just overrun with cancer cells. So that is why in some breast cancer, they have to remove a lot of the lymph nodes in the area because they're just congested with cancer cells, and we just need to get them out of there. Keep in mind on these patients, you want to be really careful about doing stuff like blood pressure, getting a blood pressure on that side, because you will cause further congestion of the lymph that's trying to get back up to the heart. So if you do have a breast cancer patient and they say, don't use this arm, this is why. Try not. Now, if they have had breast cancer on both sides, it's best bet there. You're going to have to get a blood pressure in some way. All right, what are lymph nodes and nodules? Basically, these are masses of lymphatic tissue. Lymph nodes are bigger. They're about 10 to 20 millimeters long, and lymph nodules are a little bit smaller. Well, I'm going to say a whole lot smaller. So these are just masses of lymphatic tissue. I'm going to show a close-up of what's going on in these. They contained fixed macrophages. Those are white blood cells that eat pathogens and debris. So if some sort of infection comes through or some sort of toxin, the macrophages, remember macro is big, phage means eat. These are large eaters. They're going to destroy and eat them. There's also fixed plasma cells in the lymph nodes, and that is where antibodies are produced. In your throat, well, actually, I'll talk about that in a minute. You actually have three paired groups of lymph nodes, and they are where we said earlier. In your neck, that's cervical, in your armpit area, which is axillary, and then in your groin area, which is inguinal. So that's three paired groups of these larger lymph nodes. So that's just the location. So here's a diagram of a lymph node. So over here on the left, you see the afferent vessel up here at the top where my cursor is. Afferent means it's coming into, remember A before E. So you have a number of these lymph vessels coming into a lymph node. And in here you have some B cells and plasma cells, which are infection fighting. And so the lymph travels through here. Bad stuff is caught up in here and destroyed. And then the lymph continues out the bottom, which is the efferent vessel, A before E. So this is now leaving and it's going up the lymph system, like I said, back into the heart eventually. So there's a lot of things going in here. You have the production of some white blood cells. You also have the trapping of pathogens and so forth in here for destruction. We want to destroy those guys. If you go into the doctor for your checkup or if you're sick, they're going to check for swollen glands. So usually they feel your neck and they say swallow while they're feeling your neck. Well, they're feeling the cervical lymph nodes in your neck and seeing if they are swollen. If they are swollen, that means they're very active because they're actively fighting an infection. Hodgkin's disease, you've probably heard of, basically is a type of cancer in the lymphocytes. So the lymphocytes, which are a type of white blood cell, they overproduce and they get large and they clog up your lymph nodes, and they basically just killed my cat. Okay. Yes. Pets can get lymphoma, which is cancer of the lymphocytes. And Siamese are especially bad at getting it. And that's who she was. She was a Siamese. So, and humans can get it too. Right? Dogs, I don't know, but there are certain breeds that tend to get this worse than others, just like hip dysplasia. You know, German Shepherds are real prone to hip dysplasia. Here is a swollen lymph node. Look at that. That's a pretty serious infection right there. I mean, not only could you feel it, but you could see it. So I bet if you did some blood work on this patient, you would see that the white count was pretty high because this patient is fighting off some kind of infection. All right, lymph nodules. Now, these are a little smaller than lymph nodes. You have them all over your body in the epithelium. Any place you have mucous membranes. Those are routes of entry into the body, so bad stuff can get in there. So we better have some lymph nodules ready to get rid of those invaders. Now, we have some in our small intestine, some lymph nodules. They're called Peyer's patches. We also have them in our neck or our throat, and those are called tonsils. They're located in the pharynx. You have 3 sets of tonsils: palatine tonsils, adenoid tonsils. Adenoids are really tonsils, and lingual tonsils also. So you have 3 pairs. I'll show you where they are here in just a second. But yeah, those are all considered lymph nodules. And let me get over here and let's see where they are. Okay. So right up here in the nasopharynx, there's the pharyngeal tonsil, also known as the adenoid. The palatine tonsil is right over the uvula, and then the lingual tonsil is on the back of the tongue. And like I said, they're in pairs. So when I was a child, you would get tonsillitis and they would take your tonsils out. These days, they don't like to take the tonsils out. They found out that this does remove part of your immune system when you take the tonsils out. So they tend to try to treat tonsillitis and not take out these structures. However, when the tonsils get this big, they got to go. All right, what happens as I mentioned earlier is when you're fighting an infection, or in this case with tonsillitis, kids get infection after infection after infection in their tonsils. It's just like multiple ear infections. It gets to the point where the tonsils and the adenoids just get clogged. They are so clogged with pathogens that the pathogens are just in there reproducing. They've totally overwhelmed the white blood cells and the immune system. So they're actually a haven of production of bacteria. So that's when they go, you know what, let's just take these out. And like I said, we get a little tonsillitis at my age. They would just yank them all out. I don't have tonsils anymore. Has this affected my immune system? I don't know. But I know the pediatricians are like, no, you know, that's an important part of your immune system. Let's try to leave them if we can. All right, so now you know what the lymph system does as far as lymph fluid. Well, let's talk about the thymus gland. Thymus gland, you might mention this in the endocrine system. This is a gland we don't talk about too much for a reason. Now, this is inferior to the thyroid gland in your neck, and it's actually under your sternum. So it kind of extends under the sternum in the infant and child. It hangs out from under the sternum. After puberty, it starts shrinking because it's no longer needed. And then as an adult, your thymus gland is little, tiny, and shriveled up. Now, I'm talking about the thymus gland here. All right, so what does it do? In the infant and child, T cells, which are a type of lymphocyte, a type of white blood cell that are made in your red bone marrow, and I'll show you a chart here in a minute on that. But T cells, which are made there, go to the thymus gland to mature. And they're our first defense against infection, T cells. So they migrate from red bone marrow where they're made, and then they go to the thymus to mature, and then they're released. Now, you have pretty much mature immunity by the age of 2, and by the age of 2, this doesn't happen anymore. You have enough T cells circulating in your bloodstream that they can just reproduce by cell division. So you can see you really need the thymus when you're young, but as you get older, there's no more need for it because your T cells are dividing on their own. They don't have to go somewhere and mature. So we still have a thymus. It just really doesn't do much at this point, and it does shrink from not being used. Here is a picture of the thymus. I'm going to say this is a thymus probably in a pediatric patient. This is pretty big over here on your left. Like I said, it's lies under the sternum. Okay. So how are blood cells made? In red bone marrow, your red bone marrow produces these things called hemocytoblasts. Hemocytoblasts are pretty cool because they can develop into any kind of blood cell. So in the immune system, they go to the thymus and then they basically turn into T cells. So all blood cells come from an original blood cell that can divide and turn into white blood cells, red blood cells, platelets, whatever. One type of blood cell can turn, stem cell can turn into any kind of blood cell. All right, let's look at the spleen. The spleen is a big part of your immune system. Not only does it produce lymphocytes, which is, again, a type of white blood cell or leukocyte, and it also stores monocytes and erythrocytes. It contains something called fixed plasma cells that produce antibodies. I'll talk about that more in a second. And the spleen also contains fixed macrophages. Now, when I say fixed, I'm talking about they live there. They don't travel through the bloodstream. They actually live there. They have a home and a purpose. Macrophage, again, means big eater. So those macrophages that live in your spleen, they eat pathogens and they destroy old, tired, worn out red blood cells. There's no nucleus, there's no DNA, so red blood cells can't reproduce. They can't just divide and create more. If you got a nucleus in there, it decreases the surface area. And also the shape of the red blood cell, it's sort of biconcave. It's larger, kind of like increases surface area like the alveoli we were talking about. All right. So after, and if you think about red blood cells, when they go through capillaries, they're going through a little tiny tube that is barely larger than they are. So as a red blood cell travels through the capillary, it gets beat up. It's actually having to squeeze through the capillary. So they get beat up pretty bad. And they only live 120 days. And as they come through the bloodstream and they come through the spleen, the fixed macrophages grab them and they eat them. They kill them off. But what happens to some of the components of red blood cells is pretty cool. The bilirubin, which is the colorant that makes red blood cells red, it goes to the liver and it gets put into bile. It actually gives bile its green color, which then goes to the gallbladder, which gets dumped into your small intestine. So bilirubin, here in Texas, we call it Billy Bob Rubin because that's just how we roll down here. Bilirubin is ultimately what makes poop brown. And so this is a function of the liver. If you have a patient with an infectious disease that is affecting their liver, and their liver is not forming bilirubin and excreting bilirubin because the liver's sick, then your poo will become a light clay color. So that's one of the signs of somebody that say has hepatitis or liver cirrhosis, because the liver's just not doing its job. So anyone that you think has hepatitis, you ask them, what color is your poo? And they say it's a really light gray color. Okay, we got a problem. We've got some liver failure here. Now, we're going to recycle the iron that is in a red blood cell. That is sent back to the red bone marrow for recycling, and the iron is put into new red blood cells. You can't get rid of iron. This is why you don't want a child to eat a whole bottle of Flintstones vitamins with iron. They will have iron toxicity, and then they end up going and getting dialysis to get that out. We can't excrete iron. You don't want to take too much iron. So these are all the things that the spleen is responsible for. And here I have a spleen. This cross section, this is the posterior part of the body. There's the vertebra. Here's the spleen back here. I think that says stomach. Anyway, so the spleen is kind of a little more posterior than it is anterior. And it is a dark red color, just like the liver, just like the kidneys, because it has a huge amount of blood flow in it. And of course, it's one of the big bleeders in abdominal trauma, just like the liver is, just like the kidneys are. You can live without your spleen. All right, so let's talk about immunity. Okay, we're fixing to talk about the most difficult thing in this topic here in just a minute. But overall, we're going to talk about immunity, the difference between humoral and cell-mediated. We're going to talk about B cells, T cells, the types of immunity we have, and then we'll discuss how vaccines work, which you guys already know that. So what is immunity? Immunity is the ability to destroy germs or pathogens or any other kind of foreign material that wants to enter our body. Generally, we're discussing all this in terms of infectious diseases. So immunity basically is protection from the world around us. We have fungus and bacteria and viruses and all kinds of stuff. So how well can we fight this off? And we just had this crammed down our throat a few years ago when COVID was big, discussing immunity and vaccinations and herd immunity and epidemiology. I think we learned more about it than we ever really wanted to know. So what kind of protection does the human body have from pathogens or germs? First of all, you have anatomical barriers. Your skin is your biggest anatomical barrier from stuff getting in. Your mucous membranes, we talked about that a little bit. And here's the deal on mucous membranes. You guys talked about how they're in the GI system, the respiratory system, the nose is respiratory, mouth is respiratory and GI, and then also genitourinary. That's a good way to describe all that other stuff. Mucus traps stuff, and those are potential entry points into the body, as you mentioned. And so that's where we like to have a lot of lymph nodules to trap any bad thing that's trying to enter there. As you know, if you get food poisoning, one way that your body tries to get rid of it is by puking it out or diarrhea or both. We're going to sit on the toilet with a garbage can because we're doing both at the same time. Mucous membranes, say in the respiratory system. How do you get crap out of your lungs? Well, you cough it out because it's trapped in there and you'll cough it out. So that's one way to get rid of that. The second thing that our body does is it has the inflammation response. It's short term. Everybody has had the inflammation response. This has to do with swelling and redness and itching that we will talk about shortly. So as far as inflammation, even if you are allergic to pollen or smoke particles, you will have an inflammation response. Some people worse than others. And then the third protection is the immune response. This is a long time protection. It's very specific to certain viruses or bacteria. It usually involves getting a vaccination. It also has to do with, have you had that disease? You know, if you've had a certain disease, then you're said to be immune to it. You can't get it again because your body built up a lot of antibodies. Not always true. All right. So up at the top, we've got the physical barriers. The other thing is your skin surface is fairly acidic, and acidic is good. It kind of retards the growth of pathogens. So that's another thing. And then in the middle, there's your phagocytes. Okay, those are your white blood cells that tend to eat and dissolve pathogens and crud. And then at the bottom, we have things like T cells that we're going to talk about that actually are killer cells, and they will go and destroy an invader. All right, so let's talk about inflammation. Everybody's had inflammation. You'll recognize this. This is just injury to the cell. This is just, I cut my hand, or a bee stung me, or just some kind of injury. So what happens? Well, first of all, you have mast cell degranulation. Okay, what mast cells do is they release histamine, heparin, and some prostaglandins. All right. Histamine, let's talk about heparin first. Heparin is kind of a blood thinner. It makes your blood thin so that it flows through faster, so that we can get white blood cells to the area quicker. Then you have some prostaglandins. Those are locally produced hormones, and they attract white blood cells. So when they dump out these chemicals that we call chemoattractants, okay, it's like perfume. Oh, there must be a female around here. I smell perfume, you know, that kind of thing. Or Axe. I could try to get my husband to not buy Axe. I said, honey, I think that's for like junior high guys. And he likes it. I'm like, okay, whatever. But anyway, with these chemoattractants, the white blood cells, they kind of perk up their ears and they go, oh, we're needed somewhere. So they will travel to the area where there's some sort of insult. Chemotaxis, so that what they're doing is what's called chemotaxis. They move to the area where they are needed. So you can, if you want to remember that, think about a taxi moves you. So chemotaxis. So the neutrophils, which are the first line of defense, when they smell those chemoattractants in some area of the body, they will go there so they can help. Vessels dilate and become leaky so that the white blood cells can get out of the bloodstream into the area that's damaged or attacked. So that's why you get swelling. You get all that fluid, that interstitial fluid in there. Plasma systems are activated if needed, and that is for clotting, say if you have a laceration or abrasion or something like that. And then the last thing that happens in this cascade of things is the rebuilding of the damaged area and removal of waste products like dead germs, or any other kind of waste products that may be in there. You know, venom, if it's a bee sting, for example. So these are all the things that contribute to inflammation. So what do we see when we have cellular injury? We see, as we discussed, vasodilation. So the area gets red. Vascular permeability. The vessels get leaky, so we get swelling. Cellular infiltration, thrombosis, which is clots, okay, if we have bleeding, and stimulation of nerve endings. All of this stuff stimulates and irritates the nerve endings, which causes itching. So at the bottom, if you know your Latin, the area has rubor, dolor, tumor, and color. It's red, painful, swollen, and warm. So, of course, when we do our head-to-toe patient assessment, DCAP, BTLS, these are some of the things we're looking for. And if you've ever had an injury or a bee sting or whatever, you know that you have these four things. It's red, it's painful, it's swollen, and it's warm. And then it will start itching as it heals because those nerve endings are irritated. Here is a flow chart that pretty much describes what I just said. Again, if you want to take a look at this, if you like flow charts, this kind of maps it out for you. And here is another diagram that pretty much discusses the same thing. All right, I'm going to talk about, we've already talked about inflammation, but I do want to talk about interferons. Okay, interferons up at the top, these are glycoproteins. They are released by lymphocytes that have been activated, and also macrophages and virus-infected cells. Interferons, the reason I bring this up is interferons are released in the case of viruses, including cancer, to help fight off the virus. So if you ever hear that, you know what that does. Complement system, I'm going to mention that again, but this is complement. What it does is it surrounds a pathogen and kills it and breaks it down. Complement also attracts other white blood cells. And then last but not least on this page is fever. Fever is really important in the inflammation response and healing. I looked up on the bottom here, body temperature rising above 37.2 centigrade. And I looked up last night, that's 98.96 Fahrenheit. That's not very much above normal body temp, but that's all it takes to kind of help the immune system here. So let's talk about what fever does. Fever is actually a good thing. We are so quick to try to get rid of fever, but it does serve a purpose. So it jacks up your body temperature a little bit. And because it jacks up your temperature a little bit, it takes your body a little while to get to that temperature after your hypothalamus resets the temp. So that's why when you are getting sick, you have the chills. Your thermostat has been set up a couple of degrees, but your body's not there yet. So you have a tendency to have the chills when you're getting sick. And that is why when you're getting well, you start sweating because your hypothalamus goes, okay, the bacteria is gone. Let's set the body temperature back to normal, but your body is still 100 degrees. So it's hot and you sweat. So sweating is always a good sign that you're getting better. But what does fever do? It reduces the amount of iron in the blood, which inhibits reproduction of bacteria and fungus, which is a good thing. Fever, just getting the temperature up a little bit, increases phagocytosis. That's where your white blood cells, neutrophils, and monocytes are killing and eating the invader. So that increases that. And fever also increases the function of killer cells. They kill better and faster if your body temp is up a little. Now, fever can get so high that it's dangerous and uncomfortable. And I know that pediatricians don't even say to give your kids children's Tylenol until their fever gets up to a certain number, which is higher than what it would be for adults. And so the idea is let the fever do its job. You know, now it will get to a point where it makes you feel really, really bad. And that's when you should probably start thinking about your Tylenol or whatever. But a little bit of fever is not going to hurt you, and it's going to help you get better quicker. Now, if you or I had 102 temperature, we would feel really bad. We would be really sick. But in kids, not so much. Okay, I mean, they're going to feel bad, but it's not as concerning. There's a really good article about this, Let Fever Do Its Job. And I just put the information to this research article up here for you if you ever feel that you need to read it. You know, some of these things are available on the internet, but on some of them you have to go into the UCLA library and get them that way. A lot of the research articles. You know, when people talk about research, I hear all these people on social media going, well, I researched this. No, you didn't. You looked on Wikipedia. That's not research. I'm talking about scientific research like what you're looking at right here. So sorry, that's not research. Okay, I'll be a research snob about that. All right, let's talk about antigens. Now, antigens they're okay things. Okay, they are just chemical markers that identify human cells and other kinds of substances. All right, your own body has antigens that mark every single cell in your body as yours. All right, so it's kind of like having a flag, carrying a flag that identifies you as, say, coming from a particular country or a particular state or whatever. All right, so antigens are not bad things. But everybody has a different antigen. So everybody's cells and tissues have antigens. And if that antigen, there's also antigens in stuff like pollen and grass. There's antigens in these substances, penicillin and strawberries and bees and peanuts. So a lot of substances have antigens. Let's say your immune system is sensitive to one of these antigens, like strawberries, and it causes you some distress, like itching and your eyes are watering. We would call it an allergen. So antigens are a good thing. They mark you as you. It's when other antigens get into your system that it causes a problem because your body goes, no, that doesn't belong here. I've got to do something about it. So that's what an antigen is. Now we're going to talk about two types of immunity. One is cell-mediated and one is called antibody-mediated. I think the antibody is also called humoral. Now, they work similarly. Both of them, both types of immunity, recognize and destroy foreign antigens or invaders. So they basically do the same job, they just do it a little bit differently. So this is pretty much going to be test bait for you. So we're going to try to make this as simple as possible. So let's talk about what is an antigen. Okay, that's a substance, of course, that your body recognizes as foreign, and that's going to begin the immune response. So look very closely at this. I'm going to go back over it with pictures. All right. Cell-mediated immunity involves T lymphocytes. Okay, those are T cells. They kill and destroy invaders. Antibody-mediated immunity also has T lymphocytes, but it also produces B lymphocytes and antibodies. There's your difference right there. Antibody-mediated has antibodies. We'll talk about what that is shortly. Another difference, cell-mediated immunity defends against pathogens inside living cells. And you'll see that in viruses and funguses and also malignant cells, so cancer cells. So that is who defends against that. Now, antibody, on the other hand, mediated immunity provides defense against any kind of pathogens that are located outside the cells, usually in some kind of body fluid. Could be interstitial fluid. Interstitial is between the cells. Okay, so these are the two main differences between cell-mediated and antibody-mediated. All right, let me see if I have a picture for this. I do. Okay, now don't go too crazy trying to write all this down or memorize it, but I just wanted to show you what happens in cell-mediated immunity. All right, you have some sort of foreign antigen enter your body. You have helper T cells and macrophages they're the first ones to recognize the foreign antigen, I'm sorry. So the helper T cells, they will start dividing and they become three types of T cells: memory T cells, cytotoxic, or otherwise known as killer T cells, and suppressor T cells. So we've got this invader coming in our body, and the T cells really kind of hop to it and start arming themselves. Macrophages are getting ready to eat the invader. All right, so what happens here? The memory T cells do exactly what it sounds like. They remember the antigen. And if that antigen or invader or pathogen ever comes in the body again, they will become active quicker. It's kind of like a sheriff putting up a wanted poster. So everybody goes, oh yeah, that's the bad guy. So if I see the bad guy, I'm going to go attack him really quick. So the memory T cells remember the invader. The cytotoxic T cells are the ones who actually kill off the invader. They chemically destroy him by punching a hole in the cell membrane and killing him, just like a SWAT team. So that's what the cytotoxic T cells do. Now, we talked about this a second ago. They also produce cytokines that attract other white blood cells there to the area. Remember, they put out this perfume or this smell, this chemical, and that will attract other white blood cells to come to the area. We need some help up here. And then, last but not least, that third type of T cell, those are suppressor T cells. They stop the immune response once that antigen has been destroyed. Okay, so if I go back to my other slide, the three T cells I talked about here, I told you specifically what they do here. Now, your killer T cells or the cytotoxic T cells, those are the ones that actually destroy the antigens. Now, this is all cell-mediated immunity. One thing that we didn't see here are any antibodies. Okay, so cell-mediated immunity doesn't make antibodies. That's going to be antibody-mediated. Now, when we have an invader, what antibody-mediated does is you have the helper T cells and B cells who both recognize the antigen. The helper T cell will present it to the B cell, and the B cell forms memory cells, and plasma cells that make antibodies for the antigens. So this is the production of antibodies. And basically what the antibodies are going to do is they're going to hang around in your system. And the next time this invader comes, it's going to be attacked faster and more effectively because these antibodies are going to recognize it. And this is what you get when you get vaccinations. So your body builds up antibodies and then they fight off the infection faster before it knocks you down. Okay, so we have the presence of antibodies in antibody mediated. So what's an antibody? This is a protein that your cells produce that attach themselves to foreign antigens and they label them as invaders to be killed. So over here on the left, here's your little antibody. It is sensitized to the antigens in shellfish. So this little antibody is hanging out, and if it happens to see some shellfish come in, like this guy, it's going to mark it. Notice it's marked the flag to be destroyed. And then your killer T cells and so forth will come and destroy it. So this is kind of a bigger memory system than cell-mediated immunity. Now, you can have, sometimes you have to have booster shots for stuff to increase your antibodies. We have to have a shot really every year for the flu. One reason is because the flu changes. But they told us when we first got our hepatitis B shots as healthcare providers that they weren't sure, but they thought it would last a lifetime. I had my Hep B shot, I want to say in 1986. And not too long ago, I went to the doctor and I said, would you check my titer on my hepatitis B? And it's still good. So I don't know how many years that is, but that's a lot of years. That's 40, almost 40 years, I think. So sometimes your vaccinations last a long time, perhaps a lifetime, and sometimes not. You have to get boosters for this. All right, does everybody understand what antibodies do? They don't actually kill the invader, they just mark it for destruction. Now, I'm going to go back and let's talk about something. The first time you were exposed to, let's say, peanuts, you're not going to have this reaction. You're not going to have an allergic reaction, because you haven't had time to produce antibodies for peanuts. But after the first exposure, you have antibodies who recognize peanuts if they get in your system, and it's that second exposure, or third, or fourth, or fifth, or whatever, that you're going to have that allergic reaction. The first time, no, because you don't have the antibodies. And what is an allergic reaction anyway? It's kind of a immune response that's in overkill, or overdrive, I should say. All right, we're still talking about antibody mediated. Once the antigen is marked by the antibody, so here it is marked, these plasma proteins called complement, they come and kill it, as you can see down here. And then again, you have the suppressor T cells, just like you did in the other one, that stop the reaction once the pathogen is under control. If the suppressor T cells don't do this, then you will have an autoimmune response. Again, this is something they're looking at. What causes autoimmune responses? How do we stop it? Because it does cause a lot of diseases. Now, complement, besides killing the pathogen, complement also stands guard in case more invaders show up. And also when the invaders are killed, they'll call in the big eaters or the macrophages to clean up the mess. So you see there's a lot of similarity between cell-mediated and antibody. All right, antibodies have many jobs. So they promote inflammation. We've talked about what inflammation does for us. Promotes destruction or phagocytosis. It activates complement. We just looked at complement here. That's the guys who actually kill it and then stand guard and call for the cleanup crew. Antibodies also stimulate apoptosis, where we talked about that. That's cell death. They also cause cells to clump together, which is called agglutination. And the clumping of cells prevents any more invaders from coming in. It's kind of like the wall. And they also inactivate pathogens or toxins. So this is just kind of a list of all the things that antibodies do. Now, what's the proportion of T cells to B cells? Remember, we see the B cells in antibody mediated immunity. About 75% of your circulating lymphocyte are T cells. Remember, those are kind of killer cells. They do other jobs as well. And only 25% are B cells. So if you were looking for a proportion, there you go. We have mostly T cells. There's some reason I put this slide up, but I could only guess what it is. Okay. Here is a flow chart showing you cell mediated on the left and antibody mediated on the right. That's kind of a nice quick explanation if you want to look at that later. All right, let's see here. Okay, you know what? This is really kind of a review. Lymphocytes, you have B and T. Those are specialized cells to destroy and label foreign bodies. Antigens are chemical markers that identify cells. We talked about that before. And antibodies are proteins that mark invaders for destruction. So this slide is basically just a review. And here is another flow chart covering the same information. And I don't know why I have an antigen up here and an antibody. I don't know why I have these pictures, but let's talk about some of the antibodies. There are different classifications of antibodies. There's a couple here I want you to know. So you've got IgG, IgA, MD, and E. Okay, so it tells you in the second column where these antibodies are located most of the time and what they do over there on the right. So the two I want you to know. First of all, IgM. It's located in your bloodstream, and that's a good thing because floating around in your bloodstream is a really good way to get from point A to point B. So to get to the area where you're needed. IgM is the first type of or class of antibody to respond. It's parked right there on the 405. It's ready to get on the entrance ramp. Okay. So it is the one that is first to respond or produce first during an infection. Okay, that's what you need to know about IgM. The next one I want you to know is IgE. It's located in mast cells or basophils, which are types of white blood cells. And this is the one that's important in allergic reaction. Mast cells release histamine. And histamine is the substance that causes people the most problem in an allergic reaction. So IgE is the one that you see in allergic reactions. So those are the two I really want you to know on this chart. Okay. So we're talking about like primary versus a secondary immune response. Um, Primary response, your first exposure to some sort of allergen is going to have kind of a slow response of IgM. And then your second response, usually because of the presence of antibodies, you're going to have a very fast increased response. One of the things to keep in mind when you're responding to somebody who's having an allergic reaction, how fast did this come on? Generally, the faster it comes on, it's the worse it's going to be. If, for example, she took some penicillin at bedtime, which was say 11 o'clock, and she wakes up at 4 in the morning with hives, that's a pretty slow onset. But if your patient ate some butter pecan ice cream 5 minutes ago, and by the time you get there, they're already wheezing and they have hives, that's going to be a bad reaction. So you want to kind of get an idea of how long ago were you exposed to this, because that is going to have a lot to do with the quickness of your response and also what you do. Is this a Benadryl situation, or is this an Epi and Benadryl and everything else we have in the toolbox? So do ask those questions. All right, so we've already talked about this. I'm going to skip over that. And here's just a picture of what you see in an allergic reaction. I'm going to skip over that. I told you I had a lot of stuff, extra stuff here, so you can take a look. Like I said, if you learn better by looking at one picture than another, great. Here are some factors in a just general immune response. You have two categories, external and internal. So external would be what caused this problem. Was it trauma? Was it a disease? Was it pollution? And internal factors would be the patient's age. Obviously, if they are very young or very old, they're going to have probably a worse response. Gender, guess what? Women have 1.7 times more T cells than males. Why? I don't know. You know, it may be because we're the child bearers. I do not know. And nutrition. Anybody with poor nutrition is going to have a worsened or weaker immune response. And that also includes hydration as well. Okay. Now, this is kind of tricky. We have different types of acquired immunity. How do we get immune to a certain pathogen? All right. There's several, and I have a flow chart, and I have this, so we'll go through this. All right. First of all, you have genetic immunity, and that's going to be on the very top. Certain living things don't get certain diseases. For example, people don't get oak tree wilt. Okay, just genetically, we can't get oak tree wilt. Oak trees can't get rabies. And that's just because of the genetic disposition. That's just innate. That's just how we are. That's in our genes and so forth. Now, there is another type of innate acquired immunity called natural acquired. Let me explain it. I can't think of anything in current medicine, but I can discuss this. Back in the old days when there was smallpox, all right, when smallpox was going around, they noticed that milkmaids, these women who milk cows for a living, didn't get smallpox. So why was that? Well, they were exposed by milking cows to cowpox. And there are some similarities between cowpox and smallpox. So they had been exposed to cowpox for so long that when smallpox came along, they already had an immunity to it just because of that other exposure. So that's called natural acquired immunity. I can't think, like I said, I can't think of anything now that equates to this, except maybe bird flu. I'm thinking that people who work with birds don't get bird flu because they're just exposed to it all the time. I'm not sure. Don't quote me on that. All right. Now, the next category in blue there is naturally acquired immunity. All right. You got it naturally. Active would be after you got exposed to a disease. For example, you got the chicken pox and now you're immune to any more cases of the chicken pox. That would be active version of naturally acquired immunity. Passive naturally acquired would be if you got antibodies from an external source, like maternal breast milk. So in the early infancy, the baby got antibodies from mom via the breast milk, or the antibodies crossed the placenta to the fetus. So there is a maternal immunity. And then the last category in blue is called artificially acquired, and that usually has to do with a needle of some type. So active artificially acquired is if you went down and you got a shot, let's say you got vaccinated against hepatitis B. So what happens is you get dead or weakened hepatitis B germs injected into your body, and your body goes, holy cow, that's an invader, and your body makes antibodies against it. So if you're ever exposed again, your antibodies are going to mark that hep B, and your body will knock it out before you get infected with it. So that's vaccination, the artificially acquired. And there's one more type. It's called, it's considered passive artificially acquired. And this is where you don't make your own antibodies, but you get them from an external source. All right. Rabies shots were this way, but let's talk about hepatitis A. Hepatitis A is an oral fecal transmission. Generally, here's what happens. Okay. I don't want to say In-N-Out Burger because I love In-N-Out Burger. Let's go with McDonald's. Okay. If you like McDonald's, I apologize. But you have a worker at McDonald's who's making sandwiches. Your worker's got hepatitis A, but they don't really know it because it's a mild case. They take a break, and on the break, they go to the bathroom and they poop. And when they wipe, they get a little bit of it on their hand, and they don't wash their hands. So they come back and they make you a hamburger. And guess what? You end up with hepatitis A. All right. So what happens if it's determined you have hep A? You see why I didn't want to say In-N-Out Burger, because I'm sort of a traitor here in Texas. Texas has a burger chain called Whataburger. And there's a big, once we got In-N-Out Burger down here, there's a big fight. Whataburger is a Texas chain. And now it's about half and half. Half of us prefer In-N-Out Burger and half the people prefer Whataburger. So we're at fisticuffs about this. Anyway, so if you got hepatitis A, you would go get a gamma globulin shot. And what they are giving you is antibodies that were made by some other creature or whatever against hep A. So they would give you a shot, and then the antibodies would mark that hep A, and then you could get rid of it. Okay, so these are antibodies from an external source, not that your own body made. It used to be the same with rabies shots. If you were exposed to an animal that potentially had rabies, you would go get a shot of rabies antibodies. This, I think, is hard to understand. I like this flow chart. This is still difficult because there's just a lot of stuff. I think this flow chart is a little more complicated than we just talked about, but I did type some stuff in at the bottom that might make it have more sense. I guarantee you will get a test question on this. It'll give you an example and ask what type of immunity it is, or it might give you the type of immunity and then you have to choose in multiple choice which one is the example. Okay, that flow chart's pretty tricky. Also look at the one in your book. That might be a little bit easier to understand. But I did want to warn you about that. And here is another way that it's written, which actually this one's pretty simple. This might be a lot easier to study from right here. Let's talk about vaccines. Obviously, these are administered to prevent disease. They cause you to produce antibodies that will recognize the disease if it comes in later on at some point. So what do vaccines have in them? They have what's called an immune system friendly antigen. This is an antigen. It may be a bacteria, maybe virus, whatever, that has been killed or weakened. So they don't know, you don't get injected with live germs. All we have to do is kill them or weaken them to the point where they can't do anything. And that's enough to cause you to make antibodies against them. A lot of people say, well, I don't get a flu shot because it gives me the flu. Well, that's crap. Okay. Maybe 1 in 500,000 cases. Now, it may give you some of the symptoms of the flu. When I was forced to get COVID shots, it made me so sick, every single one of them. I got the chills, I got fever, I got all of the signs and symptoms, lasted 12 hours. So it does not cause the disease, but it may give you some of the symptoms. My father would never get a flu shot. He says, it gives me the flu. No, Dad, it makes you feel bad for about a day. It's okay. A vaccine takes the place of your first exposure to a pathogen. All right. And then you develop antibodies, and then the second and subsequent exposures, you're able to fight it off before it takes over your body. And vaccines simulate the production of antibodies and memory cells. We've already talked about that. So down at the bottom, there are some vaccines. I bet you guys have had to take a lot of these because of your profession. One of the most important ones you can take is Tdap. That's tetanus, because we're always cutting ourselves. Diphtheria and pertussis. Now, pertussis is whooping cough. A lot of adults have this and don't know it. They just have this cough and they don't know why they're coughing. The problem is if you transmit this to an infant or a young child, especially a baby who's premature or someone who has a crummy immune system, you could actually kill them. Pertussis can actually kill them. So if you are around children in your job, or you have young children, or you're fixing to have a child, or you have young grandchildren, you probably need that Tdap. Oh, let's talk about autism and vaccine. There is still, in scientific research, no correlation between vaccines, including I can't say this, thimerosal, thank you. Thimerosal and autism. Thimerosal is I think it's like a mercury-based substance that makes up the liquid in a vaccine. So they've done tests on that. And who says this anyway? Well, you can go to any of the organizations, but I thought this was interesting. The Autism Science Foundation says there is no correlation. Did you guys know that this all started many, many years ago in England, where a physician whose child was having a birthday party followed 25 children that were at the party and determined that vaccines caused autism. That study, that so-called study, was so flawed. I think he got denounced and he lost his medical license for that. But that's what got everybody all upset. Do we know 100%? No. Do we know 100% what causes SIDS? No. But we know more than we used to know. And the reading I was doing showed that autism can be caused by one or the more of the following. The more genetic mutations. Now, a baby might have 1 or 2 genetic mutations, and that's not as bad as having 18. So if you have genetic mutations, the more, that's going to make them more prone. Exposure to toxins, the age of the parents. I was very surprised to read this, and also maternal medications, which could be also read as exposure to toxins. Okay, so right now, this is all they got. This is tricky, but we actually have more. When you talk about causes, we have more now on what causes SIDS than we did 20 years ago, 40 years ago. So we are making progress, but this is the latest scientific research. This is not Wikipedia right here. This is scientific research. So we're going to talk about germs. Okay, as far as microorganisms, that term has to do with things that can only be seen through a microscope. And 5 categories, even though it depends on what book you read, some give you 4 categories, some give you 7. Your 5 general categories of microorganisms that cause disease are protozoa, fungus, parasites, virus, and bacteria. And you won't see this list always this way, because a lot of times protozoa are put into one of the other categories. So I kind of listed it separately because I wanted to talk about a couple of medical conditions that you see as a result of a protozoa infection. So what are protozoa? It's really, they are a subcategory of parasites. That's what I was talking about. Usually these will be under parasites. They're single-celled, and protozoa a lot of times live in the ground or they live in water, etc. They cause these following diseases. Malaria, okay, that's a mosquito thing. We don't really see malaria over here too much. Giardia, if you are a camper or backpacker or outdoor person, you know that you just don't drink water out of a stream without boiling it or putting chemicals in it, because you will get Giardia and it will give you really bad abdominal cramps and diarrhea. And then toxoplasmosis. That's carried by cats. And you're likely to get it cleaning the cat box or the litter box out if you don't use gloves or if you don't wash your hands. This is so bad in causing birth defects that they tell pregnant women, don't clean the kitty litter box. Have somebody else do it while you're pregnant. So that's where that comes from. Fungus. Funguses are plant-like organisms, but they don't make chlorophyll. So if you think about mushrooms, that is a type of fungus that we are putting on our pizza, and they're delicious, right? But here's some non-delicious ones. Ringworm is a type of fungus, and it can cause the following 3 things. Tinea pedis, if you get it on the feet, it's called athlete's foot. Tinea cruris is what causes jock itch. And you can also get nail fungus. But keep in mind, all 3 of these are a form of ringworm. And ringworms are not worms, they're funguses. Not sure how they got the name, but they kind of tend to show themselves as circles or circular. So maybe that's how they got the name ringworm, but they're not worms. Another fungus is Candida albicans, which is a yeast infection. Now, listen, we have this fungus all over our body all the time. It typically lives in our gut, lives in our mouth. The problem with candida is if it overgrows, if you have an overgrowth of it, it can cause problems. So if you have it in your mouth, you can get thrush, which is a disease where you get white patches inside your mouth and it's painful and so forth. Or you can get the yeast infection, that women sometimes complain of. They have yeast in that area, in the vaginal area all the time, but an overgrowth can cause what we call a yeast infection. And men, you can get yeast infections too. And you can get them in all kinds of parts of your body. They're pretty easy to get rid of. They like sort of moist, dark, warm areas. So any place you have that, you're likely to get it. Parasites. This is some kind of organism that lives on or in a host and gets its food at the expense of that host. So we have a couple of categories here. We have endoparasites, which are inside the body. And mainly we're talking about worms. Helminth is a worm. So we're talking about tapeworm, pinworm, that kind of thing. We don't have worms like they have worms in other continents and countries. There are some worms that people run around barefoot. I think they're called guinea worms. People run around barefoot and the worms get in the bottom of their feet and they get in their legs and grow. And then they come out of the skin and they're like inches long and they just burrow out of the skin. And, our ER doctors here don't see a lot of that. But sometimes if somebody comes into the country, they'll be confronted with guinea worm. Look that up. It's really gross, guinea worms. So the ectoparasites, they're something that lives on the outside of us, and that includes any kind of lice, mites, fleas, ticks, bed bugs. Now, bed bugs are not microscopic, but that is a type of ectoparasite. And we did have a really bad epidemic, I don't know, maybe 10, 15 years ago, of bed bugs in hotels, even good hotels. And people would put their suitcase down on their hotel floor, the bed bugs would climb in, and then they would end up taking the bed bugs home, and now their whole house is infested. We had a fire station here infested with bed bugs. What happened was the guys went out on a call to this residence, and one of the paramedics sat on the couch next to the patient to examine them and picked up bed bugs on his clothes and then took them back to the fire station. So they had to evacuate the fire station. They had to move all the guys somewhere else. They had to fumigate, and bed bugs are hard to get rid of. They require a blood meal, and they can go 18 months without blood. So it's very hard to get bed bugs. You don't want those. A lot of people say if you go to hotels, put your suitcase in the bathtub. They can't get in there. So be careful. Also check your mattress. All right, now that I've completely grossed you out, I do want to talk about scabies. Scabies is a type of mite. They're actually little spiders. And what they do is they burrow into your skin and lay eggs. And then the eggs hatch. And then the new spiders crawl out and get on your skin. So you have these little burrow marks. They're usually in a line like this, but not always. When I got them, they were in a little line like this. And the place they really showed up was in my antecubital area on the inside of my arms. How did this happen? Well, my husband, he's a paramedic as well. He went on a call where he picked up a gentleman who was not real good at hygiene. The gentleman had bare feet. My husband was on the foot end of the stretcher. When he lifted the stretcher, this is back before the battery-powered stretchers, the guy's feet were up against his shirt, his belly, because he has, a belly. And apparently the guy had scabies, and the scabies came off and got on his shirt and crawled on the inside and got on his skin. And so he brought scabies home. And then, of course, I got them. So we had to put insecticide all over us, and we had to boil all the towels and all the sheets and all our clothes and send everything to the dry cleaners. They're very, very hard to get rid of. So that's scabies. Lice is the same idea. But anyway, yeah, this is what their burrow marks look like. Okay, now let's talk about viruses. Viruses are probably the smallest pathogen. It's a very small piece of genetic information, DNA or RNA, and it's kind of in a little protective shell, so we can't fight it off as easily. Now, viruses cannot produce unless they go inside one of your cells and use your DNA to reproduce. So they go in, they use your cell, they reproduce, and then when all the new viruses come out of your cell, they rupture your cell and destroy it on the way out. So I love this. I found this last night. I wanted to put this up. What is a virus? It's like someone breaking into your house to use your kitchen. They brought their own recipe, but they need to use your dishes, measuring cups, mixer, and oven to make the recipe. And then when they leave a big mess, meaning they cause destruction of the cell or destruction of your kitchen. This is probably the best description I've ever seen of how a virus works. So there it is from myclevelandclinic.org. So they're incomplete. They're incomplete. They have to use your cells to reproduce. Now let's go to bacteria. Bacteria is a single-celled living organism, and they're very diverse. They're categorized, there's many different types. They're categorized by shape or habitat base, that's where they are found, or person base, which means they were named after whoever discovered them. Okay, so here's your categories. We'll look at some examples here in a second. I don't want to end up going too fast for you guys. All right. And I'm going to slaughter the pronunciation of a bunch of this because I never took microbiology. So I'm bad at this, and I apologize for all you biologists out there. Spirillum, that is a spiral-shaped bacteria, as you see on the top there. Coccus is spherical, so round, and they can be in any kind of formation. You could have one by itself or a big cluster of them. They can be in a line like you see in the second picture. And bacillus is rod-shaped, okay, like you see in the bottom picture. So these are pretty much, there is one that is shaped like a comma. It's not really one of the main categories, so I didn't put it up here. But that's another one that you might see. So they're generally categorized into one or the other, depending on shape. They also can be categorized by the description, how they look under the microscope. So Staphylococcus aureus, which you see on the right over there, refers to its golden color because AU, of course, is gold on the periodic chart, right? And then down at the bottom, we see Pseudomonas aeruginosa or aeruginosa. I don't know which way it's pronounced. Does anyone know? Normally, we just call it Pseudomonas. We see that a lot in nursing homes, care facilities, hospitals. It's one of those opportunistic infections. It was named after its color, which is, they called it copper rust. All right, when copper rusts, it turns kind of a blue-green color. So there it is down there at the bottom. So that's how it got its name. This is actually pretty interesting. I was thinking about this last night. You know, at this stage in my life, there's two things I never took in college, and one was microbiology, which is really interesting, and the other is organic chemistry. I took regular chemistry, but organic is fascinating. And I'm like, and I'm the age where I get free tuition in Texas. If you are a certain age, you get free tuition. So I could go take one of these courses for free. Now I'd have to pay some fees, but I could still get in there with all the 18-year-olds, right? You can also name bacteria by its habitat. So Escherichia, I guess is how you pronounce it, coli, otherwise known as E. coli. The coli refers to colon, and that's where it lives. E. coli is not a big deal. We have to have it in our colon, but the problem is when it's in our food. So in ground beef, if they grind it up and they get some of the colon contents into your ground beef, then you could get E. coli. It'll make you really sick. Enterococcus is found in intestines. So the form of that word entero indicates that's where it's found, where it lives. And the halobacterium lives in a salty environment, which includes oceans. And I think halo is the root word for that. I forgot to look that up last night. But anyway, so by habitat, and then another way you can name your bacteria is by whoever discovered you. E. coli was discovered by Theodor Escherich, I think is probably how you pronounce it. It looks like a German name, I'm not sure. Neisseria was discovered by Albert Neisser, and Salmonella typhimurium was discovered by a scientist named D.E. Salmon. That's how it got its name. So this would be just a college course in itself, the naming of all these bacteria. I thought it was pretty interesting. But generally, most of your pathogens have what's called binomial nomenclature, and this was actually in your objectives, so I want to make sure I covered it. The words are in Latin. If you speak Spanish, Italian, French, Portuguese, the Latin names are going to look very, very close because all of those Romance languages that we called came from Latin. So there's even a lot of words in English that are originally Latin, and we can kind of figure out what they are. So a lot of the names are, first of all, the genus will be capitalized, and then the species will be a lowercase. And then, of course, they usually have more names, but I just wanted to give you a couple of examples there. Staphylococcus, that is the genus, and aureus is the species, okay? Not every pathogen is named this way, but I just wanted to show you sort of generically how they get named. It's like plant species, same thing. And the animal kingdom, same thing. All right, what's an infectious disease? This is an illness or disorder caused by a pathogen. And what is a pathogen? It's basically a germ, some kind of organism that causes a disease in the categories that we just talked about. How do we spread infectious diseases? You probably remember all of this: direct contact, indirect contact, droplets, or it's airborne. I can't remember if I go through these individually. I don't, so we can talk about it here. Direct contact, sex, kissing, that sort of thing. Indirect contact, somebody with infectious disease drank out of a beer bottle, and now I'm drinking out of their beer bottle. Yes, toilet seat, potentially indirect contact. Somebody with a disease sits on the toilet seat, and now you sit on the toilet seat. So that's indirect contact. Droplets, sneezing, coughing, talking. I bet you guys know somebody who, when they talk, they just spew huge amounts of saliva. And yes, you can become infected by that. And that's why they, told us to wear a mask, because COVID is transmitted by respiratory droplets. And then there's airborne. I don't remember what year this was, But there was I can't remember, I think this is a bacterial infection that originated in the air conditioning system in the water on top of this hotel. And they had this big convention of Legionnaires. And because this was in the air conditioning system, all of this was just being spewed out in the AC, and everybody caught this Legionnaire's disease. I don't remember. I think it was maybe the 80s or 90s. It's been quite a while. But yes, you can have some pathogens that are airborne like that. So these are the major ways that we can spread infectious diseases. Common infectious disease, your objectives say list some, and I have them here. I think I spelled syphilis wrong. I just took a look at that and it looks kind of funky. But these are some common infectious diseases. You guys, as EMTs, are already familiar with these. But we also have good germs. So we have what's called normal body flora. These are good germs. These are germs that actually help us out. Some of the common locations that you will find this is on your skin, in your oral, nasal, oral, and respiratory tract, your GI tract, urogenital area. So that's urine and genital area, and conjunctiva, which is your eyes. So these are just some places where you have some good flora or good bacteria, and you don't want to get rid of. All right, so let's look at what some of them do. There's body flora on your skin, and what it does is prevent the colonization of pathogens, right? Interesting, a body flora that prevents pathogens. The body flora that resides in your mouth and your respiratory system kind of helps stabilize your immunity. You have bacteria in your GI tract that does a couple of things. First of all, it helps produce vitamin K and vitamin B and biotin, and it also helps you metabolize drugs and nutrients. Some of the flora in there prevent colonization of other pathogens. So if you have to take antibiotics for some reason, the antibiotics tend to kill off all that flora in your GI tract, and you start ending up with diarrhea. That's one of the big problems. That's one of the side effects of the antibiotics. So they always say, if you're on antibiotics, you probably should eat something that has flora in it, and yogurt's probably the number one thing to look at there. Eat yogurt, take antibiotics, eat yogurt, because it will cause diarrhea once you kill all those little guys off. In the urogenital tract, females have a vaginal flora that maintains an acidic environment in the vagina that protects against infections. So, like I said, if you get an overgrowth, you end up with a yeast infection. And conjunctiva, that flora sort of maintains the microenvironment of the eye. So you do have flora there as well. And, like I said, antibiotics are pretty bad because they're going to not just kill off the bad bacteria, but they're going to kill off the good bacteria in your body, and you will likely have side effects. Now, I'm going to leave you with this. There's these things called Demodex mites. They live in your eyelash follicles. They're good. Now, these are microscopic. You can't see them crawling around. They're there, and they eat dead cells, like flaky skin crap and sebum, which is excess oil in your hair follicle. So they live there and they reproduce. But you don't want to overgrowth of them. So what you're looking at here are not the mites, but you're looking at mite crap. So if you don't wash your face very much, or if you don't scrub your eyes occasionally, this is just mite crap. This is just excrement and all kinds of stuff. This is a really nasty one from these mites. So yeah, you got mites, and you'll never get rid of them all. They're there for a purpose. They actually help you out. But if you don't stay clean and wash your face, you're going to get this issue right here, and it's going to itch and irritate you. So I didn't put a picture of the mite up because I didn't want you to totally be freaked out, because it's a gross looking mite. Just saying. All right. What causes problems with your normal flora? Number one is overgrowth. We talked about too much yeast. You're going to get a yeast infection. Introduction of additional, like E. coli in your hamburger meat. All right, that will make you sick, even though it lives in your bowel. Any kind of injury or surgery can introduce problems. Bowel surgery is a real issue. I knew a couple of people that had that weight loss surgery where they take out feet of your intestine. And what happens if you have any leakage out of that intestine into your abdominal cavity, you're going to end up with a nasty, it could kill you, bacterial infection. And simply a skin laceration can do the same thing. Heart surgery is another problem where bacteria can get introduced. Migration from one area to another. For example, you can have pathogens that migrate from the anus to the vagina, or the anus to the urethra. And of course, anus, think about GI system, there's a lot of bacteria in there. All right. And last but not least, the disrupted immune system. Okay. I can't remember if it was in the 80s, I believe. Doctors noticed that these young men were coming into the doctor's office. Now, see, just thinking about those mites is making my eyelashes itch. Anyway, young men were coming into the doctors with this pneumocystis cystitis pneumonia, a very rare type of pneumonia, young guys. And doctors were like, what is going on? You just don't see this type of pneumonia. And it's they found out it's a type of pneumonia that people suffer when they have a depressed immune system. Of course, you see that in AIDS patients. And that's kind of how they discovered AIDS in this country, is all these guys were coming in with this certain type of pneumonia, and they were like, what is this? So anyway, that came about, I don't know, about 1986 or something like that. And there's other things that cause immunosuppression that you're just not going to get this disease unless your immune system's not working properly for whatever. If you guys have any questions about anything, there's my email address. And if I don't see you again, the best of luck on the rest of AMP and also paramedic.